Hello, everyone. I am Susan Daniel. I'm a professor in the department here, but I'm also the director of the school. And my laboratory is about 15 students and postdocs big, and we tend to study things on the biological side of chemical and biomolecular engineering. In particular, we're really interested in understanding processes that happen at or within the cell membrane. So some of the things that we study are the process of viral infection. That's probably about a third of my lab's efforts. The other part of the lab studies bacterial membranes. And so some of the applications there are trying to understand how antibiotics could work better. And then the other part of my lab studies chemical processes at cell membranes that occur inside your cells. Our aspiration there is to try to carry out the same biochemical reactions that happen inside your cells, but in little devices that we can make and control ourselves. So we like to call this a synthetic cell or a cell on a chip. In particular, we've been studying for a number of years coronavirus. Actually, before the pandemic ever occurred, we were really interested in understanding how the spike proteins worked. And so the spike protein, as you probably know from the news, is the protein or biomolecule that decorates the outside of the coronavirus. And that is what interacts with the host cell membrane. And so we're interested in understanding how that little nanomachine actually knows which cell types to bind to and how it is used by the virus to open up a little hole that allows the genome of the virus to escape the packaging of the virus itself, enter into the host cell, and then through that process replicate more viruses that then go on to bud from the cell surface and proceed to infect other cells. And so our group has really been studying a particular piece of the spike protein that's called the fusion peptide. And the fusion peptide, as the name implies, really controls that fusion process. And what's really interesting about the fusion peptide is it's just this little portion of the protein, but its job is to sort of bury itself into the host membrane so that it can start to drive open this wedge that'll eventually become this opening that allows the genome out. And so you might wonder, well, why the heck would a chemical engineer ever want to study something like that? That probably isn't what you imagine a chemical or biomolecular engineer focused on. But for us, it's really about, it's a little nanomachine and understanding how biology creates these little nanomachines that have these particular jobs that they do and do really well, and how we can learn how they're put together and if you will, reverse engineer them is kind of a fun, intriguing pursuit that we're interested in understanding better. And of course, if you understand that, then you can do all these other great things like, create different therapeutics against that process, or maybe even use that inspiration to create a new kind of particle that would help deliver genes or medicines to cells that we would like to try to help them and make them healthy again. One fun fact of our research group is that we have 7 languages spoken in it, which is pretty fun. We also have a number of married people in our lab, so families are welcome. Other fun facts are things that we like to do outside. So, for example, we spend a lot of time at our lake, which is a beautiful place to spend a nice sunny summer afternoon. We've gone hiking together. Sometimes we go camping together. We have barbecues. So not only do we work hard, we also play hard and have a good time too. Well, I can tell you what I think my approach to mentoring is, but as I tell everybody, I think you also need to talk to other people who I mentor to find out what they think about my mentoring style. But what I can tell you is the spirit of what I try to be as a mentor, and that is really thinking about this process as an apprenticeship process. So you'll come in, you'll start with our team, and you'll be guided or shepherded through the process of getting your PhD. So in the beginning, you won't know much. You'll have a steep learning curve, a lot to learn, but you'll be supported in that process. You'll be supported by me and by my team members and by other graduate students who know those techniques and tools already, who will guide you along the way. Along the midpoint, you'll start really working hard on your own projects. and your independence will be growing. And then by the end, you'll be essentially a peer of mine, right? You'll be able to guide your own research and have your own independent research ideas, and you really won't need me anymore, which is exactly the time when you will leave the nest and go on and do whatever great things you're going to do. So my style tends to be one that I believe kind of morphs along the process, right? So in the beginning, it might be more hands-on. And then as time goes on and your independence grows, you'll be more and more in the driver's seat of what you're doing and seeking me, checking in with me maybe less and less because, your confidence and abilities have grown. So yeah, I would say my mentorship style is really one of looking at this as I'm your shepherd and trying to carry you along that pathway so that when you get to the end that you're really prepared to go on and fulfill whatever research dreams that you have that you would like to pursue. What's fun about living in Ithaca is that we have these great seasons and great, opportunities to embrace those seasons and do fun things in all of them. So the summertime, we have a boat that my husband and I love to go out on, and the lake is just a real center point of being here in Ithaca. It's beautiful. It's 40 miles long. You can never, get bored of it. Water ski, swim, whatever you want to do, play with your dogs. And then we have this glorious fall season where all the colors change, and just going for hikes through these beautifully, colorful forests is something that's really indescribable. Winter comes and you could get out your snowshoes or your winter skis and go for a cross-country ski around the lake, for example, the little lake here on campus, PB Lake. That's fun to do. And then by the time you're sick of winter and ready for things to change, spring pops open, flowers bloom, and it's one of the most beautiful places you could be. So, so there's always something to look forward to in Ithaca.